Hello everyone, in this video we're going to talk about DNA replication and the central dogma. So with DNA replication, if you take a look at this picture right here, it says, sorry boss, I can only do the 5 to 3. So the idea behind this is that DNA replication can only happen in the 5 to 3 direction. So if you're not really sure about what the 5 and 3 is talking about, you should watch the other video where I explain the DNA structure and the RNA structure. So with DNA polymerase, which is an enzyme that's in charge of DNA replication, can only work in the 5 to 3 direction. It can only slide down the DNA and add new bases from 5 to 3. So there's a video right here that's really good. You should watch it. It's only 3 minutes long. Go ahead and click on it when you get a chance. It's called DNA replication 3D, I think, and it's less than 3 minutes long. So it shows you the process of DNA replication a lot better and in a more animated way. But I'm going to continue on and tell you what I expect you to know in terms of DNA replication. So when you look at this picture right here, and again, DNA is double-stranded, right? So on the DNA strand, there's actually something called the leading strand and the lagging strand. So you don't really need to know for regular biology which one is the leading strand, which one is the lagging strand. But if this one is the leading strand for one part of the DNA, this part, the opposite one will be the lagging strand. So what it's showing over here is that DNA polymerase works in opposite direction as well. Because the two DNA strands are opposite in direction, DNA polymerase obviously is going to run 5 to 3 in two opposite direction as well. Okay, so that's just something you should know about. In this experiment, you don't really need to know the details of this experiment, but you should know that DNA replication is a semi-conservative process. So what we mean by that is we have our double-stranded DNA twisted, all winding together, right? We call that the double helix. But then once the DNA unwinds, you have to add new bases. So you're going from one DNA to two DNAs, or two strands of DNA to 4 strands of DNAs together, right? So if you have these 2 strands of DNA bounded together, you first have to split them apart, and then you add the new bases. That's how you can go from 1 DNA to 2 DNA, right? There's still 2 double helices. But what you see is that in order to make a new DNA, you have to have some sort of template. Otherwise, how do you know what the code's supposed to be? So you have the DNA that are matching with each other. It has complementary base pairs. You split it apart, and based on this single-stranded code, you can add complementary base pairs, and that's how you can get 2 full DNAs. So it says newly synthesized DNA contains 1 old strand and 1 new strand of DNA, and this is called a semi-conservative process. So if you look at this picture on the bottom, you can see how this 1 old, 1 new works, right? So if this were to be, actually, this picture is a little confusing, but the idea is your new DNA actually contains 1 strand that's from the original DNA and 1 that's newly synthesized. DNA replication occurs during the interphase of a cell cycle. So if you can remember, the cell cycle contains G1, S, G2, and M phase. And G1, S, and G2 are all called the interphase because it's in between cell division. But before your cell can divide, you have to replicate the DNA, right? Otherwise, if you have the same amount of DNA and you divide, now you only have half the DNA. That doesn't work, right? So the G1S, the S phase, is called synthesis phase. And what we mean by synthesis is we're synthesizing DNA. So synthesis phase, the S phase of the interphase, is when DNA replication happens. So DNA has to unzip, right? If your DNA is double-stranded, you have to unzip it first. And remember, the 2 strands of DNA are bonded together using hydrogen bonds. So when the DNA unzips, it's unzipping the hydrogen bonds. And the enzyme that's in charge of unzipping the DNA is called helicase. Okay, so helicase is only used during DNA replication and nowhere else. It's just for DNA replication. Although during transcription, we still need to unwind the DNA. We do not use helicase. The next one is DNA replication has DNA polymerase. So DNA polymerase catalyzes the replication of the new DNA from the 5 to 3 direction. So what we mean by that is that if you look at this picture, we have this a helicase unwinding the DNA, right? It's separating the hydrogen bonds. But then we have to actually add the new bases, right? So if we add a new base right here, you already have the old A. On the other side, you would add a T. And this side, you already have the T, you would add an A. C, you would add a G. This opens, you add another G, you add another T, right? Eventually, you're going to get 2 double helices. So who actually adds the DNA bases? DNA polymerase, the enzyme called DNA polymerase, actually adds the DNA during DNA replication. And again, DNA polymerase only works from the 5 to 3 direction. There's also something called proofreading, right? If you think about writing an English essay, you have to go back and proofread your essay because even if you have an idea in your head, the way you write it out might not be exactly how you want it to be. So same thing with DNA replication, although the codes are very clear what new base is supposed to be added onto the old DNA, sometimes errors can occur, so DNA polymerase can actually go back and fix its own mistakes to minimize mutation. A mutation is a change in a DNA base by accident, okay? Sometimes a mutation could be good, it could be bad, it could be neutral, but we want to minimize those harmful effects of mutation. So again, each strand of DNA is served as a template for the new strands according to the base pairing rules. A pairs with T, C pairs with G. So if you can imagine, if we have the old strand over here, A, T, T, C, G, G, on the opposite strand, you would have T, A, A, G, C, C, right? So if you have the old strand, you know how the complementary works, the opposite strand will be made. Obviously in our cells, there's not a lot of thinkings going on. This is all being done automatically. The next thing that we want to talk about is something called telomeres. So telomeres is an extension to the end of the DNA. So something that happens during DNA replication is that every time it replicates, DNA shortens, which means every time your cell replicates its DNA and allows cell division, the DNA actually becomes shorter. So what's the problem with that? A lot of the DNA contains genes, right? And if you can remember the definition of a gene codes for a protein or a trait. So if you're shortening the DNA, the part that has the gene or that codes for a trait, that's not a good thing. Your DNA is now containing less information than it's supposed to. Then something is going to go wrong in the cell, right? And that's not great. So what do? Our cell, we can't prevent DNA from shortening. Just the DNA replication process makes it shorten every single time it replicates. So what happens? We have something called telomeres being added to the end of the DNA to protect DNA, right? So the DNA is still going to be shortened. If you look at this picture, the DNA is still getting shortened every single time it replicates. But instead of shortening the actual gene that codes for a protein or a trait, you're shortening the telomeres, which are basically useless information in terms of what it codes for. It doesn't really code for anything, but it protects the genes from being shortened. So if you look at the words, telomeres added by the enzyme telomerase, right? Telomeres, telomerase. So the enzyme is called telomerase. It protects the possible loss of genes at the end of the chromosome, right? The DNA, again, is still going to be shortened every time it replicates, but it's not. The genes contained on the DNA is not going to be affected because it's really the telomeres that get shortened. So what happens when a cell loses all of its telomeres? Then the cell dies. We actually want cells to have sort of a lifespan. We don't want cells to be able to divide forever because otherwise the cell will become cancerous, right? That's how you get tumors, is when cell division is uncontrolled. So we want control over how many times the cell can divide. So you can take a look at these review questions, which are on your review sheet as well. You should be able to answer all these questions. Moving on, we're going to give you a little overview of the central dogma. So if you're very comfortable with central dogma already, you can probably skip this part, but otherwise you can keep watching. This is kind of the basics of the central dogma, okay? So this is just the difference between DNA and RNA. You already learned that. Oh, this one's kind of funny. Amino acid, okay? So proteins. The reason why we're talking about proteins is because the central dogma is how DNA is expressed, right? Again, if you watched the last video, you would have known that I talk about how on your computer, you have the computer code that helps running the computer. But really what you see on the computer screen is not the code, right? That will be rather inconvenient. You have to interpret the code. It's not going to work out so well. So what you see is kind of the expression of the code. You're seeing what the codes are coded for. Same idea with the DNA. You have the DNA instruction in every single cell, but how is the cell actually going to run? Or how is your multicellular body going to be built, right? We have to put the proteins in charge. So the DNA is kind of in charge of giving the instructions, and proteins are the ones that actually gets the work done, which is why we're talking about amino acid, because proteins are made out of amino acid, right? So it says the function of a protein, it controls everything in an organism or the body does, including the traits. So a lot of our traits are actually built by proteins. We say that proteins are the expression of your DNA. It's the genotype of your genes, right? So we have genotype is the DNA itself. So proteins are kind of the phenotype, we say. You don't have to know that yet. But the idea is, again, the DNA is the instruction. Proteins are the ones that gets the job done. It shows what the DNA is really coded for. And then it says, but DNA cannot make proteins alone. It needs some help from another type of nucleic acid called RNA. So if you think about this, right, this is not a huge problem in prokaryotes, but in eukaryotes we have the nucleus, and DNA stays in the nucleus, and protein is made in the ribosome, which is in the cytoplasm, right? So DNA can't come out of the nucleus, and ribosome is going to stay out of the nucleus. So what do we do? How do we make this protein? So how we do make this protein is that we have the DNA code. We transcribe it into RNA code. So RNA is now holding the information that DNA has. And then the RNA will leave the nucleus of a eukaryote, get to the ribosome, and then get translated into proteins. So proteins is directly made using the information from an RNA. And again, this picture just shows you DNA replication, transcription, translation. And again, from the earlier part of the video, if you're using DNA polymerase, you have to use it for DNA replication. You can't use DNA polymerase for transcription. That doesn't work. Transcription is making RNA. Why would you use DNA polymerase, right? So protein synthesis, let's take a look. Protein synthesis is DNA sending its information to RNA, specifically messenger RNA, which is also called mRNA, to make a protein, right? Think about it. We have information in the nucleus. We have ribosome outside of the nucleus. This messenger RNA is sending the message out of the nucleus. Excuse me. When we're talking about a protein, proteins are made out of amino acid, right? So nucleotides are kind of the building, what we call the building blocks of DNA or RNA, and amino acid is one makeup of proteins. So one protein is made out of a ton of amino acids in a chain. So we have one amino acid connected to the next, connected. Between each amino acid, the type of bond that's formed is called a peptide bond. Peptide bond is a type of covalent bond, which is why we say a polymer of a protein is called a polypeptide, because it's a chain of amino acid, and a chain of amino acid is called a polypeptide. And a protein really is, you have this polypeptide, but you have to fold it into a shape before it actually functions. So we have amino acid, which is the single piece, the building block for proteins. We have polypeptide, which is a chain of amino acid. And once this polypeptide folds into a shape, that is actually a functional protein. Location. Again, proteins are made at the ribosome, which is an organelle that's either floating around the cytoplasm if you have a prokaryote or a eukaryote, or if you were to be a eukaryote, you can also have ribosome attached to the rough ER, which is what makes the rough ER rough. This picture shows you again, with DNA, we can do transcription. So how does that work? We have to unwind the DNA, and unwinding of the DNA is done by DNA. Oh my God, don't ever say that. The unwinding of the DNA in transcription is done by RNA polymerase, okay? Not helicase, not DNA polymerase. RNA polymerase gets all the jobs done for your information. So we have to unwind the DNA a little bit, otherwise RNA polymerase cannot come in. And then we add RNA bases. T matches with A. A normally supposed to match with T if it were to be a DNA, but RNA doesn't have T. So we match A with U. And then we have C matching with G, T matching with A, blah, blah, blah. And then we have G. What's the next one supposed to be? What is it supposed to be match with if we're making an mRNA? It should be C, right? And then it should be G, U, A, and A. So transcription is to make an mRNA using the information of DNA, because complementary base pair, just like how two strands of DNA can complementary base pair with each other, RNA can also complementary base pair with RNA or DNA. So during transcription, RNA bases can match base pair with the DNA template strand. Okay, so you can see how this is a template that you're using to add the RNA bases. And the coding strand, well, you don't really need to know the names of the template strand and the coding strand, but it helps, right? If you think about it, template is what you use to add things. You use it as a template. And the coding strand actually contains the same code as the mRNA. So if you see this code right here and this code are exactly the same, except for the T and the U's. Okay? And then once you make an RNA transcript, then you can take this mRNA out of the nucleus, go to the ribosome, and make a protein by connecting the correct sequences of amino acids together. This video, again, is only 3 minutes long, but it's really good. Please watch it. Please, please, please. It's only 3 minutes. It's worth your time, I promise. Now, let me talk about Central Dogma a little bit. So the Central Dogma is what we call gene expressions, right? Or the process of protein synthesis. You go from DNA to RNA to protein. DNA, RNA, protein. So you should know, if I were to ask you what is the Central Dogma, it's using the DNA code to make RNA, to construct RNA, and then you use the RNA code to construct an amino acid chain or a polypeptide or a protein. So DNA is used to make messenger RNA or mRNA. DNA can make other RNAs as well, but messenger RNA is the only type of RNA that can be used to make proteins. Right? It's the only kind of RNA that consists of information that tells you how to make this protein. So you can think of the mRNA kind of like a recipe book. You have this recipe book, you read this recipe, and now you can make the food, okay? And protein is kind of like the food. So mRNA for short, using RNA polymerase. So RNA polymerase does two things. It unwinds the DNA and it adds the RNA bases. So if you can look right here, this is the RNA polymerase, and it's adding the bases of RNA. So no T's allowed, we're making RNA, and it's unwinding the DNA. This happens in the nucleus if we were to be talking about a eukaryote. If we're talking about a prokaryote, this happens in the cytoplasm. The mRNA then leaves the nucleus and goes to the ribosome to make a protein. And mRNA is the instruction for making the protein. If you look at this picture, we have our single-stranded mRNA. It's not very happy about being the messenger boy, but you gotta be the messenger boy.